biology. So we're still talking about protein structure, as you can see, but now we're going to think about how these large and sometimes very complex three-dimensional shapes can be formed by this long polymer, this polypeptide chain. How does it fold up into these precise shapes? Because whether we're talking about the spike protein of the coronavirus with all its alpha helical domains, and up here the beta pleated sheet domains, those domains have to be arranged next to each other in this very precise orientation in order for the spike protein to do what it does, in order for it to have the right shape to recognize the surface proteins on a cell that it infects, and to have the right shape to actually what it does is it changes its shape and kind of slides apart so that it can latch onto a cell so that the virus can get into the cell. And it's the same thing with trypsin here. It has to have that particular shape. This is a space-filling model, but you can see that you can't even tell that that's a long polymer when it's all folded up like this. But what you're seeing is certain amino acids are more on the surface, some are going to be on the inside, and it's going to be folded into this three-dimensional form that now allows trypsin to act as a digestive enzyme. And what it does is it allows it to digest proteins, as it turns out. So quickly here, let's, not too quickly, let's take a quick review of what we were talking about in the previous lecture about primary structure. Recall we're talking about amino acids linked together by peptide bonds. And so this is what we call the primary structure, or the sequence of amino acids in the polypeptide. And because there are 20 different amino acids and they can be linked together in any order, you can have thousands of different possible combinations of amino acids in the protein. What determines which amino acids are there and what order they're in? Well, that's determined by the genes. It's determined by the information in DNA, as we'll find out later in the semester. But in any case, that's held together by those peptide bonds, which are covalent bonds. And then recall that the secondary structure is when that long polypeptide is either coiled up or folded up in some very regular form, like an alpha helix or a beta pleated sheet here. And both of those types of structures, and there's another type of structure called a turn that we didn't really talk about, but all of those structures are stabilized by hydrogen bonds between the peptide bond groups. So this is the initial folding of the protein. You see a lot of that in proteins. Most of the structure of a protein is going to be wound up in either alpha helical form or beta pleated sheet form, because that's going to be the most stable form for the polypeptide to take. All right, now just as a review, recall that peptide bond is formed by condensation or a dehydration synthesis between two amino acids. We remove a molecule of water from across the carboxyl group and the amino group here, and dehydration synthesis produces this peptide bond. And that peptide bond then is a covalent linkage. So it's a very strong linkage. Right? You can only break that by the reverse reaction, right, of hydrolysis, and that is going to require not only the specific reaction to happen, but it requires an enzyme to do it. An enzyme, for instance, like that trypsin, and that's going to break apart what we have here is a dipeptide into two amino acids. So my point here is that is a covalent bond that can only be broken by a chemical reaction. Now, for the secondary structures, those are also held together pretty tightly, and that's because you have, although they're only hydrogen bonds, which are not chemical bonds and not nearly as strong as a covalent bond in an aqueous solution, but there are so many of them, and they're at regular intervals throughout, whether it's a beta pleated sheet structure or an alpha helical form. Lots and lots of hydrogen bonds, and the attractive force of those bonds kind of adds up when you have a lot of them in regularly spaced intervals. All right. So now we want to talk about the tertiary structure of a protein. The tertiary or third level structure of a protein is going to be this irregular folding of the polypeptide due to the R group interactions now. It's the first time we've seen the R groups really do anything yet. So this is going to be more complicated. It's going to be stabilized by 5 different types of interactions. So we saw with the primary structure, it was just peptide bonds, that's covalent. With secondary structure, it was just hydrogen bonds. But now we have lots of different interactions all working together to hold the protein in a particular form, or what we call a configuration. So these interactions are things that you already know about. So we'll see the effect of hydrogen bonding, basically, in the hydrophobic effect. Van der Waals attractions occurring among nonpolar groups. Hydrogen bonds will form between polar groups and between polar groups in water. We have ionic attractions that are going to occur between charged R groups. And then I put this in parentheses because to a minor degree, there will be one type of covalent bond, but it will be rare. So those are all the different interactions that are going to be involved in holding this tertiary structure together. So what I want to do is go through them one by one and consider their contribution. So the first is the hydrophobic effect. And this is the same effect that we saw that basically placed all things like fats together in a little bubble with water surrounding it, right? It's the exclusion of anything nonpolar into the interior, in this case of the tertiary structure, because of all the hydrogen bonds that occur between water molecules. So water molecules will just hydrogen bond to themselves because they are so polar, very sticky. So anything that can't hydrogen bond with water is going to be just ignored by water and pushed away, and they end up together. So I can try drawing a polypeptide chain. I'm just going to draw it as a line because, I like to draw everything as a line. So I'm just going to draw this. It's not folded up yet. I'm not putting any secondary structures in it because it's just too hard to draw, but assume that they're in there. So I'm just showing the amino terminus and the carboxyl terminus. So I better label that line as the polypeptide. Otherwise, that line could be anything, right? I've used a line to show everything from cellulose to starch and everything else. So say you have sticking out from this polypeptide backbone various R groups. If those R groups are nonpolar R groups, in other words, if those R groups contain methyl groups, so like the hydrocarbon I'm drawing here, I'm just drawing the R group part, and then just methyl groups there, those are nonpolar groups, right? Carbon and hydrogen bond is nonpolar. It's not going to be able to hydrogen bond with anything. So there's water out there on the outside. Consider this the outside of the polypeptide. And those water molecules are going to be hydrogen bonding with other water molecules. So what that does is it forces all of the nonpolar groups away from the water, essentially, as the water arranges itself around this structure. So if you have things like alanine, which has a methyl group, or valine here, which has a larger hydrocarbon, the R groups of those amino acids are going to be found more to the interior of the polypeptide as it folds. Now, once they get in there and they're pushed close together, then you can have some interaction between them, right? And you know what that would be if you have nonpolar R groups, a nonpolar group of some type, right? There's only one way they can really interact, and they're only going to do it if they're very close together already, and that's by van der Waals interactions. So the van der Waals interactions will help stabilize this structure, but they're not going to be the main, they're not going to be what forces the hydrocarbons to the inside. That's because of hydrogen bonds between water. So they're just being excluded together, and then kind of as a secondary effect, they can be, I spelled van der Waals wrong there. As a secondary effect, they can then interact here. We'll put it like this. So the nonpolar R groups are going to interact by van der Waals. So I'm going to include that in our list of interactions. The first being the hydrophobic effect is really just due to hydrogen bonding. And then as a second effect, a second interaction, I'll include the nonpolar groups interacting by the van der Waals interactions or attractions or forces. Okay, so those are our first two effects. Now keep in mind, if you remember that chart we had of the amino acids, there are a lot of nonpolar amino acids, 9 of them, compared to out of all the 20. So this is quite an important effect in how proteins are going to fold. Another thing we have to consider, though, are the hydrogen bonds that can form between polar R groups or between polar R groups in water. So we know what those polar groups would contain, right? Any kind of functional group that has the nitrogen-hydrogen, the hydroxyl, or carbon with oxygen, those are those 3 polar bonds that you were supposed to memorize. So if you see those in a functional group, you know that it's possible that that group could hydrogen bond with water. So, or it could hydrogen bond with other polar groups. So it's both ways. So let me attempt another one of these drawings. Again, I'm going to put just an abbreviation for the amino terminus or the N terminus of the polypeptide. And it's folded up, and as it's folding, now probably the first thing that makes it start to fold would be that hydrophobic effect, but once you have certain parts of the polypeptide in close proximity to other parts, if you have R groups that are sticking out that have polar groups, they could interact. So I can't draw this in 3 dimensions or with anywhere near the complexity we're talking about here, but I can just throw in a little polar R group here. So that's a group that has a hydroxyl group, that's the amino acid serine. And I'll put a fancier one here, fancier being slightly more complex, an R group that has both a carbonyl type, carbon double bonded with oxygen, and an amino group. So this is, this first one is serine with just the single hydroxyl group, and then this other one is asparagine. And I want to remind you that you don't have to memorize the names of these 20 amino acids or their structures. This is not something you have to do for this class. I'm giving you examples. And the more you see them, the more you become familiar with them. That's got to be a good thing. All right, so we can have a hydrogen bond formed between the partial negative end of asparagine's functional group and the partial positive end of serine's functional group. So that's a hydrogen bond between polar R groups, and that will help hold that part of the protein together. So remember, out here around this protein is in an aqueous solution in the cell, there's water. So those water molecules, I'll draw them out this time, those are going to be hydrogen bonding with other water molecules. That's what they do. Now, if there are R groups there that are sticking towards the outside, though, then those water molecules could hydrogen bond with them. So here's a polar group that could hydrogen bond with one water, and then another polar group. I just did that. I put that to the wrong part of the water. That's not a real hydrogen bond at all, is it? Well, I can fix that. So the idea being that we have various hydrogen bonds that can form to the outside of the polypeptide as well. Here, fixed. It's like magic. So I did want to show that if you have a hydroxyl type functional group and an R group, then the partial positive of the hydrogen is going to associate with partial negative of the oxygen in water. So what you end up with is a solvation shell, right? You get water molecules arranging themselves around the polar and, as we'll see, the charged R groups to the outside of the protein, whereas all the more hydrophobic R groups will be to the inside, but also near the surface and different parts of the protein will have these interactions between polar R groups that will hold certain parts of the protein together. So all these interactions are working together to form this shape. Another type of interaction that's important are ionic interactions. So electrostatic interactions between charged R groups. So which R groups are charged, that could have a charge, a positive or negative charge? Those that have the acidic and basic functional groups, because remember, they can be protonated or ionized. So think about the carboxyl groups and amino groups, right? So let's draw another. I'm drawing another one of these polypeptides. And think about what we mean by an ionic interaction between a charged R group. I don't know why I made that so big, but I did. All right, make a big R group then. So an example of one of the R groups that can be charged would be the R group, would be the amino acid lysine, which has an amino group that usually at pH 7, but certainly at acidic pH, would carry a positive charge, right? It can be protonated. Another one that's acidic is basic, like lysine, is asparagine. So now if you have another somewhere else on this polypeptide chain, you could have another amino acid that has a different R group. Say it has a carboxyl group, and a carboxyl group, as you know, can be ionized. It can act as an acid and lose that hydrogen, and now you have an anion. You have a full negative charge. So this is going to be a very strong attraction. So the one I've drawn here as an example, because it's easy to draw, is aspartate. And of course, that is the anion of aspartic acid. And generally at pH 7, it's going to be negatively charged. So I put little dots to show this particular interaction. This is going to be a stronger attraction than just a hydrogen bond, right? Because now we have a full positive and full negative charge. This ionic interaction, though, is not called an ionic bond because it's not making a true salt, right? It's still dissolved in water. But what we call it is a salt bridge, because it will bridge the different parts of the polypeptide chain and hold those two parts together. And you can have lots of these different salt bridge type interactions in some proteins, and they help hold the protein together very tightly into a particular shape. You can also have charged R groups on the surface of the protein, and those would, of course, interact with water. So if you have an R group that is positively charged, something like glycine, it would interact with the partial negative on the oxygen, and that would attract water to it quite strongly, right? And then if you have a negative charge, that's going to have an electrostatic attraction to the partial positive of the water molecules, polar bonds. So these are electrostatic interactions either between a full positive and full negative, so +1 and -1 charges, that's a very strong attraction, or in the case of the groups on the outside, it could be a full positive and a partial negative, and a negative anion and a partial positive water. Either of those is going to be stronger than just a hydrogen bond, right? Because the charges are higher. So salt bridges are going to be a little stronger in holding the protein together compared to the hydrogen bonds. But there aren't that many of them if you think about the number of potentially charged R groups. There's 4 or 5 depending on the pH, basically. So there aren't that many opportunities, and some proteins may not have a lot of charged R groups in them. Some may have a lot. The other thing that I don't show here is that sometimes we'll see this later on when we talk about enzymes. It's possible for some proteins to have ions and other non-protein parts that are folded up with them, and the ions then can act as ligands, basically, and help hold the protein together as well. And we'll discuss that idea when we talk about enzymes. So the last type of interaction that is important in some proteins is a covalent bond. Now this is a covalent bond between R groups. We're not talking about the peptide bond group, and this is actually pretty rare. You don't find it in every type of protein structure. So this is called a disulfide bridge, and it only occurs between 2 cysteines. And a cysteine is a particular amino acid, right? So it's the cysteine amino acid. Its R group has a sulfhydryl as its functional group. Remember, a sulfhydryl is a sulfur and hydrogen. So there are only 2 amino acids that have sulfur in them. One of them is cysteine, the other is methionine, but only this one can form this salt bridge. So this is very different than those other attractions because it's a covalent bond. This is a chemical bond, and the only way to form a chemical bond is by a chemical reaction. So I'm going to draw 2 pictures here. The first one, again, just drawing our polypeptide chain as it's folding up, and all these other interactions have helped fold it into a particular shape. Usually the formation of the disulfide bridge is kind of later in the process after everything's folded into its shape. And what's happened is you have 2 cysteines that have been brought close together. So you can see I've got one cysteine here with its sulfhydryl group and another one I'm going to put on the other side. And then a chemical reaction has to be catalyzed in order to bind those together. So I'll just draw cysteine and cysteine here. So remember, the sulfhydryl is the name of the group that has the sulfur and hydrogen in it. Now, if you take an upper division course in biochemistry, you'll probably learn a lot about how these sulfhydryl groups are formed and how they can be broken. But for now, just understand that we're talking about a chemical reaction that has to occur to link those 2 parts of the protein together permanently. So I'm just going to redraw it as they've been brought, the parts of the polypeptide have been brought a little closer together by this interaction, this attraction, this bond. And so I'm not I'm just going to show, this is just an illustration, a cartoon illustration, not where each atom would actually be linked, right? So we can see this is the disulfide bridge here, disulfide because the two sulfurs are covalently linked to each other. That is a disulfide bridge. And keep in mind, it's a covalent bond, which means it is a permanent chemical bond. In terms of the relative strengths of the interactions we've been talking about, this would be the strongest, right? This one is not going to be broken down just by heating the protein or by any of the denaturation treatments you might have tried in lab or looked at being tried in lab. So it's very strong, but it's rare. And we'll see that where you find those disulfide bridges is often in proteins that are going to be used in or found in places where they have very either you have really unusual conditions, like some type of bacteria, say, that live in boiling hot springs. They would definitely have to have a lot of disulfide bridges. Or in certain compartments, even within our cells, there are certain compartments that have a very acidic pH, and the disulfide bridge would help hold them together. So we'll see examples of that as we look at different types of proteins and what they do. So just as a summary, this is a textbook cartoon showing the same type of thing. You can see they put a few of the alpha helices in there. And it is true that when you look at the structures, you'll find that most of the length of the polypeptide chain is going to be first folded up into either alpha helix or a beta pleated sheet or this turn type structure, because those are the most thermodynamically stable structures that a polypeptide chain can take. So we have the hydrogen bond here between a couple of polar groups. And we have another one between a couple of other polar groups down here. There'll be a lot of hydrogen bonds holding parts of the protein together. Here's some hydrophobic interactions, what I'm calling the hydrophobic effect that puts those together, and then secondarily the van der Waals interaction that helps stabilize them together. And then a disulfide bond, that permanent linkage between parts of the polypeptide. And here's an ionic. It's not an ionic bond. I would prefer use the term salt bridge for that, or an ionic attraction, right? So that you have a full positive charge and full negative charge of an anion and a cation being held together. So those are all the R group interactions that determine the tertiary structure. It is possible, by the way, sometimes for the peptide bond groups and even the termini of the ends of the polypeptide to be involved somewhat in these R group interactions, but it's mainly the R group interactions. So when you see a structure like this, and this is again the spike protein from the COVID-19 corona, and what you're seeing, what they've shown here is the space-filling model, and they've taken, as we'll see, the spike protein is actually a trimer, so there are 3 separate polypeptides stuck together. And this is one of them here, and you can see there's some beta-pleated sheet region up here, a lot of alpha helical strands down here, so you don't see very much of it that's what they call just open, free polypeptide. Oh, there's a little bit of it. Random coil, they call that. So, but what we'll see is these various sections of the protein, because of the interactions between the R groups, sticking out of those alpha helices, for instance, it's going to hold those in a very precise shape, but it's also going to allow the protein to function in a particular way. For instance, this section of the protein with these alpha helices, those alpha helices can slide and move and change the shape of this protein so that it grabs onto the surface of the cell. So we'll see that certain amino acids, as you can see here, certain R groups are more likely to be on the surface, some are more likely to be into the interior. And the spike protein, as an example, is a little bit different than some of the enzyme we've been looking at, because part of it is going to be stuck into a lipid. So part of it's surrounded by water and part of it isn't. And so we'll talk about how membrane proteins will have a slightly different rules for their tertiary structures. But most proteins will have both. Now, there are some protein tertiary structures that may be almost entirely alpha helices, and there are even some that are almost entirely beta pleated sheets. This one's got a little bit of alpha helix down here, alpha helix. So sometimes that's just the shape of that protein is more based on those structures, but it generally will have one or the other. And then we'll see some It's the same type of thing. You have another tertiary structure here with mostly beta-pleated sheets, one with mostly alpha helices, and then here's one with a lot of disulfide bonds or disulfide bridges. And this is, I know it looks a little funny with the yellow there, the disulfide bridges. It's just a conventional kind of a diagrammatic way to show a disulfide bond, a disulfide bridge. And so you'll see those in some of the structures that we encounter. So, there you go. So what we've done so far is we've looked at the primary structure, which is just the sequence of amino acids. We've looked at the secondary structures, the alpha helix and the beta-pleated sheet, how they all fold together and are superimposed into this tertiary structure, so the final folded form. And then what we're going to do next time is we're going to do two things in the next lecture. I'm going to talk about how these tertiary structures, how we can determine the shape of the structure after it folds. So there are some methods that have been used for years to figure out what is the shape, what is the three-dimensional shape of that structure. Some of those I've been showing you. How do they figure out what the shape of all those folds and coils in the spike protein, for instance? How is that determined? And also another aspect of protein research is can you predict this tertiary structure, this final folded form, from the primary structure, just knowing the sequence of amino acids? And the answer is not very easily. For most proteins, it's not that simple because there are thousands of different possible configurations that a polypeptide chain could take. So the idea with trying to predict it is to figure out which would be the lowest energy, the most energetically favorable form, the configuration for it, for all these R groups to be twisted into. So we'll talk about that a little bit. And then finally, we'll talk about another level of organization in proteins called the quaternary structure. This fourth level is when you have more than one protein held together. And we'll talk about that as well. So these fancy structures with their ribbons and coils are very nice to look at on a picture, but it's not something that's certainly easy to draw, right? So if you want to, whoops, what happened here? If you want to show an illustration, the illustration going. The way I usually draw things, if I was drawing on the board, I would be showing things drawn as lines, right? So a squiggly line here representing the polypeptide, and I've got it drawn into some sections of alpha helices because those are easier to draw than beta-pleated sheets. So this is just the polypeptide with some secondary structure. And as we'll see when we talk about how these structures are formed or how polypeptides are formed, those structures tend to form right away and rather spontaneously based on their sequence. But then, once those structures are formed, then the entire tertiary structure is formed by these R group interactions. So consider now that the alpha helices and in the random coil, the turns, they're all going to be kind of interacting with each other because we have all these R groups that are sticking out from the coil, right? And those R groups are going to interact by those 5 different types of interactions that I mentioned so that everything gets folded up into a particular shape. I'm not making a very nice shape here, but just you get the idea. Those R groups interacting, and now we have a folded form. So when we have a folded polypeptide, it's folded up like this, and it's in its final form, and now it's in its functional state, as we'll see, with all the tertiary structure intact, right? All the R group interactions intact. This is when we usually refer to a polypeptide as a protein. Now, an unfolded polypeptide is often also referred to as a protein, but functionally the protein is when it's in this form. It's a little ambiguity in how we use those words, but it's fine. So now this protein can be unfolded. Right? So anytime those R group interactions are disrupted, because that's the only thing that's holding it into that folded form, if the R groups are disrupted by anything, so by heating or agitation or any of those other treatments that you looked at in lab, right? A change in the pH perhaps. Sometimes heavy metals get into the structure that disrupt the ionic interactions. Anything that can change those interactions can unfold the protein. because that's all that keeps it folded. Now, a denaturation like this, it's gonna unfold the polypeptide, so you lose the tertiary structure. But generally, you don't lose the secondary structure in most cases. So when we have an unfolded peptide, we just refer to that as a denatured protein. That's what we mean by a denatured protein. It still has the polypeptide. We haven't broken apart the amino acids. It's still an intact polypeptide. It's just unfolded, right? And I just want to make note that I left the alpha helices in there because usually the secondary structures remain, and that's just because you have all those regular repeated hydrogen bonds over and over and over in those regular intervals. And although each one of those hydrogen bonds is relatively weak attraction, they kind of add up along the length of that helix to hold it together more tightly. Whereas with the R group interactions, you just have a few hydrogen bonds here, a couple of ionic interactions over here. It's not such a regular structure, so it can be denatured. So we'll talk a little bit more about denaturation and how it can happen. In I'll go over that probably in the lab recap for the second lab for the biomolecules lab, and then, as I said in the next lecture, we will move on